In this lecture, we'll talk about edible vaccines. What is vaccine? So, vaccine is a biological preparation that improves immunity to a particular disease. Vaccine, it's a word derived from uses of cowpox. Cow means vacca in Latin. It vaccine contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbes, or its toxin or one of its surface protein. The agent stimulates the body immune system to recognize the foreign antigen and then destroy it and protects the body. Edward Jenner in 1796 used this vaccine in human beings, resulting in protection of human beings from smallpox. A disease antigen that stimulates the body to produce an antibody reaction but is not strong to produce the disease harmful effects. So it will induce an immune response, but it won't cause the disease, harmful effects of the disease. So that's vaccine. Vaccine is a biological preparation that establishes or improves immunity to a particular disease. So there can be two types of vaccine. Prophylactic vaccine to prevent the effects of future infection by any natural or a wild pathogen. So that is prophylactic. And therapeutic means it's to treat any particular disease. It can be used for treating a disease like cancer vaccines. So vaccines against cancer. So they are called therapeutic because they can cure cancer. So what is an ideal vaccine? What should be the main criteria to consider a vaccine an ideal one? It shouldn't be toxic or pathogenic. This is the one important one. It shouldn't cause any toxicity in humans. Low levels of side effects. Side effects should be minimal, should be very, very low, or it should not contaminate the environment, should be safe for environment. It shouldn't cause problems in individuals. Techniques of vaccine should be simple, so delivery of the vaccine should be simple, and it should be cheap so that it can be used for larger for the mass for a large number of the human population. So what is edible vaccine actually? In the edible vaccine, transgenic plants are used as vaccine production system. So here we are using transgenic plants. The gene encoding antigen of bacterial and viral pathogens can be expressed in plants in a form in which they retain their native immunogenic properties. So here actually we are in for making edible vaccines. We can take antigen. We can take genes encoding antigens of bacterial or viral pathogen and then express in plants, which can be then consumed by human form, and then they can protect us from the diseases. Initially thought to be useful only for preventing infectious disease, it has also found application in prevention autoimmune diseases, birth control, cancer therapy, and other disease. Edible vaccines are currently being developed for a number of human and animal diseases. So why do we need a edible vaccine? Oral vaccine, so it's a needle free. Okay, so you don't have to inject. Most of the vaccines it has to be injected. Particularly the kids don't like being injected. So oral vaccine. So these edible vaccines they are used as oral vaccines. So the oral vaccine provide mucosal immunity at various sites by secreting antibodies. Do not need to worry about reuse, misuse, and lack of sterilization, thus low risk of infection. So this is the advantage of edible vaccine. They are needle free. And they are very cheap. Estimated cost of $0.005 to grow antigen for one dose of hepatitis B vaccine in unprocessed form. So it's very cheap. Cost cost of production is also less, and also it will be very cheap for the vaccination. Administering oral vaccine would require little or no training at all. So you don't need a trained experts or personnel for giving this vaccine to the patient. Storage, another advantage of these edible vaccine is the storage. It can be stored for long period of time. They are highly heat stable, do not require cold chain maintenance. For other vaccines, you require ice or the cold chain maintenance is needed. But in this case, edible vaccine, they are heat stable, so we don't require any cold chain facility. If the local native crop of a particular area is engineered to produce the vaccine, then the need for transportation and distribution can be eliminated. If, like for particular area, uh, local crop can be identified, and then they can be that particular crop can be engineered to produce the vaccine, then we can avoid the cost of transportation and distribution. So and it can be delivered very fast to the patient, to the needy person, and they are highly safe also. Most importantly, they trigger the immune at the mucosal surface as the mouth, which is the body's first line of defense. Needs no purification, because here the and this is edible vaccine. The plant will material will be consumed, and there is no need of purification, so that's why they are also cheap also, because downstream processing is now not required in case of edible vaccine. Edible vaccine activate both mucosal and systemic immunity. So these are some of the advantages of using edible vaccine. So this is the same thing. The developing world's mostly deadly disease, cholera, rotavirus, and E. coli infection, to name few, enter the body through the gastrointestinal tract. A vaccine that is ingested may actually provide the best protection, because it mimics the natural route of infection. But like particularly for diseases like cholera, rotavirus, and E. coli infection, they enter the body through the GI tract. So a vaccine that is ingested may actually provide the best protection, because it mimics the natural route of the infection. So that's why edible vaccine may work much better in these diseases. So how does edible vaccine work? So we'll just go through mechanism of action. The goal of oral vaccine is to stimulate the mucosal and systemic immunity against pathogen. Edible vaccine, when taken orally, undergo the mastication process, and the majority of plant cell degradation occurs in the intestine as a result of action of digestive or bacterial enzymes on edible vaccine. Once it is degraded, then Peyer's patches are Peyer's patches in our body are an enriched source of IgA producing plasma cells, and have the potential to populate mucosal tissues and serve as mucosal immune effector site. The breakdown of the edible vaccine near Peyer's patches, consisting of 30 to 40 lymphoid nodule on the outer surface of intestine and contain follicles. So these follicles act as site from which antigen penetrate the intestinal epithelium, so thereby accumulating antigen within organized lymphoid structures. After that, the antigen then comes in contact with M cells. These are called microfold cells. M cell passes the antigen to macrophages and the B cells. Then these B cells activate the T cell to provide immune response. In this way, the immunity is activated by the edible vaccine. So this is the mechanism of its action. So Peyer patches play a very important role here. So this picture shows the same thing here. You can see here, these are the M cells here, microfold cells. So M cell passes, so when the antigen from the vaccine, edible vaccine, so you can see the antigen which is present in the in our food, so it's broken down, and it then taken up by the M cells, Peyer's patches. M cell passes the antigen to the macrophage and the B cells. So here is the B cells here, and here the macrophage cells. These macrophages display pieces of the antigen to helper T cells. So here is the helper T. So here you can see this macrophage is presenting antigen to the T helper cells. Okay, so they're presenting to helper T cells. T cells stimulate B cells. Now T cells will then stimulate the B cells and seek out antigen at the distant site. So once these B cells gets activated, they produce the antibodies against this particular antigen, and then it protects the from the particular disease. So for activated B cells make and release antibodies able to neutralize the antigen. So this is how the edible vaccine work. So now when a disease agent appears now, so that was the scenario when edible vaccine was taken up by the body, consumed by the person. And now suppose now and virus enters our body. So here is the infected cells, here again the M cells. So now we have once uh, once an antibodies from the edible vaccine is able to create the memory. This is the initial response. So they are able to create memory T cells in our body. So these because of these T cells are helper T cells are created because of the edible vaccine. So when the virus infects the body, these memory helper T cells produce cytotoxic T cells to attack then they activate actually basically the cytotoxic T cells, and these cytotoxic cells then will kill the infected cells. Again, memory helper T cells will stimulate antibody secretion. So basically these memory helper T cells will then activate memory B cells to produce antibodies, and then these antibodies will quickly neutralize the invading virus. So this is how the edible vaccine can protect the human from the further infection by any virus or bacteria with help of by generating helper T cells, memory helper T cells. So how to develop an edible vaccine? So there are two ways. In one case, the entire structural gene is inserted into plant transformation vector between 5' 3 regular element. This will allow the transcription and accumulation of encoding sequence in the plant. So this is one way. Second, epitope within the antigen. So here the whole, in one case, the whole complete structural gene is inserted. In second case, epitope, just the more highly immunogenic part of an antigen can be identified, and then that particular epitope can be then used to construct gene by fusion with a core protein gene from plant virus like TM, tobacco mosaic virus, and others. So this is the chart shows the production of edible vaccine. So you develop the vector, introduce the vector into the plant cells using agrobacterium or microprojectile, and put this into the leaf. You get the after antibiotic selection you get the transgenic leaf disc and new plant with the antigenic prion. So now these plants will express the antigen of the your choice from either bacterial antigen or viral antigen, and then protect us from the various bacterial or viral disease. So this is now a edible vaccine now. So this is like example of in a carrot actually. So method for transformation, this is like one most commonly used is agrobacter mediated gene transfer, or one can use macroprojectile bombardment method also, or electroporation method also. So these are the commonly used method for introducing genes into plant. So this is agrobacterium tumefacin mediated method. So this is bacterial cells. You identify the first step is to identify highly immunogenic antigen in from this particular bacteria, E. coli or Salmonella or any. And then introduce that into agrobacterium, and then put this into and you cut the leaves, expose leaf to the bacteria carrying an antigen gene and an antibiotic resistance gene, allow bacteria to deliver the genes into the leaf cells. So here this is the agrobacterium here, in which the antigen gene of a particular pathogen is introduced into agrobacterium, and then it infects the leaf disc. Okay, and the DNA will DNA carrying the antigen gene will enter the plant cell and get integrated in the whole plant genome. And because of the antibiotic resistance gene present in this vector, you can select for the recombinant callus. Okay, so the callus which will survive in the antibiotic medium, they will be the recombinant one. And then once the callus is developed, you induce shoot formation and the root formation, and then you get the edible vaccine. Like suppose if you have selected the plant potatoes, you have the now potatoes can be used as a edible vaccine now. So there are several species of plants can be used for developing edible vaccine, like tobacco, potato, banana, tomato, rice, lettuce, soybean, alfalfa. All of these, muskmelon, carrot, peanuts, wheat, corn, all of these plants can be used for developing edible vaccine. So each plant species has different advantages. So tobacco, it's a good model for evaluating recombinant proteins, low cost preserving system. Potato dominated clinicals. Most of the uh, foreign genes, bacterial or viral genes has been introduced into potato, and these are many transgenic potatoes are undergoing clinical trials. So this can be easily manipulated, easily propagated, and stored for longer period without refrigeration. So as you know, potatoes can be stored at room temperature for long period of time. So this is the advantage of potato, can be stored for longer period. The only disadvantage of potato is using that it needs cooking, so which can denature the bacterial or viral antigens and then decrease the immunogenicity. So this is one disadvantage, but the storage is but the long storage is a good advantage for the potato. In case of banana, if you have edible vaccine using banana, it doesn't require cooking. Okay, it's inexpensive, and developing country is grown quite widely. Only thing is takes 2 to 3 years to mature. So this and it gets spoils very fast. So this is a disadvantage. You cannot store for long period of time. Tomatoes, it grows quickly, cultivated broadly, high content vitamin A. It can help actually. So these are the advantages of tomato also. So tomato is also another good source, but only thing that this also spoils very fast. Rice commonly used in baby food because of low antigenic potential, high expression protein antigen. So expression of the foreign proteins can be very high. Only one disadvantage is grow slowly. Okay. Lettuce fast growing, soybean large harvest number of times years. Okay. So these are different. So each has several advantages and disadvantages also. So based on the requirement, these plant species can be selected for developing edible vaccine. So these are some of the example where the clinical trials are going on. This is antidote toxicogenic E. coli. So this is developed by Thompson Institute USA. Accomplished the first published successful human trial in 1997. 11 volunteers fed raw transgenic potato experiment LTB. 10, 10 of these individuals developed neutralizing antibodies and 6 developed a mucosal response. So this was the clinical trial. Successfully, the neutralizing antibodies were observed in the humans. So this was again the E. coli. Norwalk virus, actually transgenic potato expressing Norwalk virus antigen showed seroconversion. 19 out of 20 people fed with transgenic potato expressing Norwalk virus antigen showed seroconversion. So antibody response was quite good. So attempts are underway to engineer bananas and powdered tomatoes expressing Norwalk virus. So this is going on. Cholera vaccine, transgenic potato with CTB genes of Vibrio cholerae was shown to be effective in mice. Early in mice, eating one potato a week for a month with periodic booster was said to provide immunity in the mice in animal models. Measles, mice fed with tobacco expressing MVH could attain antibody titers 5 times the level considered protective for the human. So the antibody response was very good in case in mice. MVH edible vaccine doesn't cause a typical measles, which may be occasionally seen with the current vaccine. So this is one advantage here. Transgenic rice, lettuce, and baby food against measles are also being developed. So lot of activities is going on for developing edible vaccine. Hepatitis B parenteral VLP could invoke specific antibodies in mice. First human trial of potato-based vaccine against hepatitis B have reported encouraging results. The amount of HBS antigen needed for one dose could be achieved in a single potato when cloned into CMV, plasmid HSV subtypes showed higher expression in roots as compared to leaf tissues of the transgenic potato. So this slide shows the status of the plant-made vaccine for veterinary purpose, for particular for controlling and diseases in the animals. So there's so lot of work is going on, and lot of, so this is Newcastle and Newcastle disease. So this disease is highly infectious, affecting domestic poultry and wild birds. NDV transmission occurs through direct contact with secretion or discharge of infected birds and contact with humides. The world's first regulatory approval for PMV was against NDV. So this already is this for in poultry, this vaccine is already approved. HN protein from NDV was expressed in tobacco cell system and found to retain the size and immunoreactivity. So this already is there in the it has been approved for Newcastle disease in poultry. This vaccine is, which is developed in tobacco, is already approved. For foot and mouth disease, this is another common disease in animals. Foot and mouth disease, FMD, is one of the most contagious viral disease of the wild ruminant and domestic animals. The causative pathogen of FMD is FMD virus. FMD is a single-stranded positive sense RNA virus possessing 4 capsid protein VP1, VP2, VP3 and VP4. So VP1 protein is critical determinant for vaccination and FMD. With the induction of VP1 noted antibody required for the immunity. So VP1 is highly immunogenic, so this it was selected. VP1 capsid protein was selected for developing edible vaccine. Studies have shown the potential of using VPN capsid protein as a subunit in potato, tobacco, and tomato. So this is vaccine is being made in using these plant species: potato, tobacco, and tomato. So this some another work actually product highly concentrated heat stable hepatitis B surface antigen in maize. So this is one work actually published in 2012. So this is for developing edible vaccine for hepatitis B. So, in case of hepatitis B virus, over 350 million people are chronically infected with hepatitis B virus worldwide. The disease burden is primarily felt in developing countries where the lack of refrigeration, of trained health professionals, and low income levels. So, here in these developing countries, edible vaccine can be of better use because you don't need cold storage facility. In developed countries, there are still large segments of the population that do not respond well to injected vaccine, like hemodialysis patient, celiac disease patient. So they are poor responders. So there is also issues in developed countries also because the poor response of the uh, other vaccines. HBsAg is a membrane-bound protein, a class of protein that are typically difficult to express in heterologous systems. So they have used a new construct. So these DNA constructs demonstrate improved accumulation of HSB antigen over previously reported materials and deliver maize grains suitable for oral vaccine that is cost-effective, heat-stable, and highly concentrated. So this now they introduce this HSB antigen in maize, and now they found out that this can be used for as an oral vaccine for prevention of hepatitis B virus infection. So they made different construct with using globulin 1 promoter, GLB1 promoter. Okay, this is HBS. This one is HSB and hepatitis B surface antigen under control of globulin 1 promoter, and there are different promoters like this extended globulin 1 promoter to basically improve the expression of hepatitis B surface antigen. These constructs were then introduced into maize germplasm using agrobacterium mediated transformation. All plants were backcrossed to non-transgenic HA2 parents, and single seeds from this backcross were analyzed by ELISA. Typically, single insertion packed with highest concentrated HB antigen selected for further breeding. So, whichever uh, plant got revealed highest concentration HB acid, so they were selected for further breeding. So basically, this picture shows the accumulation of HSB antigen, HSB antigen in different constructs. So maximum expression was seen in this particular construct. So this is the same thing. So now the this particular group then studied the effect of temperature on the vaccine. An ideal oral vaccine would maintain antigen integrity at ambient temperature over a long period of time. So it should not get degraded fast. In parenteral vaccine, HSB antigen reduced antibody titer in human population when the injected vaccine is exposed to 45 degree for one week, 37 degree for 1 month, or to ambient temperature. So the earlier vaccines actually basically they were not very stable. So when they were exposed to 45 degree for 1 week, they lost their activity. Cereal grains can stabilize protein for several years at ambient temperatures. Once you have the cereal grains like maize or rice, they can stabilize the protein for several years at ambient temperature. You can keep for several years. But the other vaccines, HSB antigens, they were not very stable. So at 37 degrees Celsius, only for 1 month they are stable, or even the ambient. So not more than 1 month. Okay, so this stability is very low, so they are highly unstable only for at 37 degree they are 1 month. So you need cold storage for long term storage. So but in the case of edible vaccine at cereal grains, particularly maize or rice, they can stabilize protein for several years at ambient temperature, so you don't need any cold storage. At room temperature it can be stored for several years. HSBNT experiment maize grains was ground and exposed for 1 week or 1 month to a range of temperature between 20 degrees Celsius 80 degrees to span and exceed a normal ambient temperature. So they took the maize grain, which was recombinant maize grain, which is expressing hepatitis B surface antigen, and then treated it to different temperature 20 degree to 80 degrees Celsius. So most assessing protein stability in maize grain for other antigens were conducted using oil oximetry by hexane treatment. Therefore, in this method, they have used two different methods: hexane treatment, supercritical fluid extraction method. So they use different method for the checking the protein stability. So here you can see here. So this is the total soluble protein in this part. Okay. So at 80 degrees Celsius, you see a lot of degradation. Okay. And in case of HSB antigen, 55 degrees Celsius, this is at 20 degrees Celsius, or this is 55 degrees Celsius. Hexane treated, or you can say here in this case of SFE treated. So this is supercritical fluid extraction method. You can see at 55 degrees Celsius also still a lot of antigen, HSB antigen was still present. It was not degraded at all. The concentration remained the same. Okay, in this case. Here it is 45.9 microgram antigen per gram of maize. Here it is at 55 also it showed 48.5. So at 55 degrees Celsius, still 55 degrees Celsius it was stable, but at 80 degrees Celsius it was degraded, HSB. So at least we can say that this protein is stable even at 55 degrees Celsius. So it means it can be stored at room temperature for longer period of time. So this is also again this is the HSB, this is the immunoblot. This is Western blot analysis. You can see the standard protein here, HSB antigen. Here you can see 20 degrees Celsius. You can see good amount of protein. 55 also you can still see the very nice protein band of HSB antigen here. But 80 degree then start getting degraded here. So this is just to show that the protein made in the maize grain present expressed in the maize grain is highly stable at 55 degrees. So basically this vaccine can be stored for a longer period of time at room temperature. Just to conclude, the high level accumulation of the hepatitis B surface antigen at mean concentration of 0.51% total soluble protein maize Steven seed using an improved version of the globulin 1 promoter. Estimated expressed in maize seeds was extremely heat stable, tolerating temperature up to 55 degrees Celsius for 1 month without degradation. So 55 degrees Celsius for 1 month without any degradation. Optimal heat stability was achieved after oil extraction with ground maize material, either by supercritical fluid extraction or hexane treatment. This study just to show that viral and bacterial proteins can be easily expressed in the plant system, and then you can be used as an edible vaccine. However, there with even like others vaccines, there safety issues will be always there. Like most probably is the allergenic and toxic potential with the plant components. So there some of the plant components that can cause allergic potential for interference, production of oral tolerance, risk of typical measles, health and environment risk of GMO as others like other transgenic plants, or other issues will be the over or misuse of these edible vaccine. And most important is public perception, how they take these transgenic plants as edible vaccine. Mostly there is in many developing countries there is a negative attitude to GMO, which can influence the vaccine acceptability in many developing countries. Legal and ethical consideration regarding products from plants with such with status like tobacco. So many people may not like to consume tobacco, to take tobacco, uh, vaccine made in particularly in tobacco. Quality control will be issue. Can antigen expression be consistent in crops? Who will control vaccine availability and production? So these are some of the issues which needs to be addressed. But still, there are several advantages of edible vaccine. Do not require administration by injection, so it can be easily given to kids. So they can, like suppose if it is in banana, they can easily have their vaccine with bananas. Possible production vaccine with low cost, so the cost of developing these vaccine is very low. Once you have once you have developed the edible vaccine, then you have to just grow it. Okay, so the production cost is very low. As do not require separation and purification of the vaccine from plant material, so there is no need of downstream purification steps here. So that's why it is very cheap. Necessary syringe and needles are not required. Economical in mass production and transportation. Easy. The best part of the edible vaccine is that it can be produced in very large amount, and then it can be easily transported because they are highly heat stable and cold storage is not required. So this is the example. This is just like to show that edible vaccine can be easily accepted by kids and others. The question is that could we one day eat plant as a drug instead of processing them? Yes, it is an attractive idea and can be possible. If right doses can be expressed in that plant with the help of food processing technology, it is maybe possible in the future.